Have you ever stopped to wonder how scientists can possibly look at all the incredible complexity in our DNA and find that one gene that might cause a disease or even hold the cure? It's a huge challenge, one of the biggest in modern biology. All right, let's get into it.
Think about it. Your body has this massive genetic blueprint. We're talking over 20,000 genes. So imagine you're a researcher comparing a healthy cell to a cancerous one. The activity levels of pretty much all those genes can change. So the question becomes, how in the world do you find the changes that actually mean something, the real signals, in that giant ocean of genetic noise?
And this challenge, it's only getting bigger. Technologies like microarrays and RNA sequencing are amazing. They give us the power to measure the activity of every single gene all at once. But what that creates is a total data deluge, a literal mountain of information that is just impossible for any human to sort through by hand. So to tackle this massive data problem, scientists needed a specialized tool, something like, a gene detective. And that right there is where a really powerful piece of software called Lemma comes into the picture. Lemma, and yeah, that's short for linear models for microarray and RNA-seq data, it is that detective. It's designed for one specific job, to sift through these enormous genetic datasets and pinpoint the genes whose activity has changed in a way that actually means something, to find the clues that really matter.
Okay, so how does this detective actually work its magic? Well, it takes the data on a pretty simple three-step journey. And if you follow these steps, you'll see exactly how we get from that giant, messy mountain of raw data to a genuine moment of scientific discovery.
So first up, it cleans the signal to get rid of all the background noise. Then it models the specific question the scientist is asking. And finally, the fun part, it finds the stars of the experiment, the genes that are really the most important. All right, let's dig into step 1, cleaning the signal. Think about it like you're tuning an old radio, right? You're twisting the dial, trying to get past all that static to hear the music clearly. Well, the raw data from a gene experiment is just like that. It's packed with static. All this technical noise that can totally drown out the real biological signal we're trying to listen to. Okay, just look at this visual transformation. It's pretty incredible. On the left, that's the raw, noisy data. See how that big cloud of black dots is all fanned out and messy? That's the static. It's almost impossible to see what's important. But after Lemma does its thing, look to the right. Wow. That cloud of noise gets squeezed together, and suddenly the real signals, those red and green dots, just pop. Those are the genes we care about, and now they stand out crystal clear. So, moving on to step 2. Once our signal is clean and clear, the scientist has to tell Lemma what the experiment was actually about. What were we looking for? This is done by creating what's basically the experiment's blueprint. It's how you translate a complex scientific question into a language the software can understand. Now, I know the term linear model can sound super intimidating, right? But honestly, this blueprint is often just a simple table, just like this one. It's called a targets file, and all it does is tell Lemma which samples are which. See here? It's just distinguishing between the normal wild-type mice and the knockout mice. That's it. This simple file is the key to translating the entire experimental question for the software. And that brings us to the final and most exciting step, finding the stars. We've got clean data, the software knows the experimental design, and now Lemma performs its analysis to find the genes that are really behaving differently between the groups. And the result is the ultimate payoff for any researcher, a top table. It is exactly what it sounds like, a clean, ranked list of the most important genes ordered from most significant to least. This table is basically a roadmap. It tells scientists exactly where to focus their attention next. So instead of being lost in a sea of 20,000 possibilities, you now have a short, prioritized list of the absolute stars of the experiment. But what really makes Lemma so incredibly powerful, especially for real-world biology experiments where you might only have a few samples, well, it has a kind of statistical superpower, a secret sauce, if you will. So here's the key. Instead of analyzing each gene all by itself, in isolation, Lemma is smarter. It looks at the behavior of all the genes at once. It literally borrows information about how genes vary across the entire dataset to make a more informed, more confident decision about each individual one. This gives it way more statistical power, which means it's much better at finding real signals and avoiding false alarms, even when an experiment is small.
So as you can see, this isn't just some dry, technical exercise. This process, going from a mountain of noisy data all the way to a clean list of top genes, this is how raw data gets turned into actual real-world scientific discovery.
And Lemma is right at the heart of some incredible breakthroughs. I mean, we're talking about understanding what makes cancer cells tick, uncovering how genes guide the development of an embryo, seeing the system-wide effects of just turning off a single gene, and even mapping the functions of different cells in our own bodies. It's huge.
It's become such a fundamental tool that a 2015 paper in a major journal just stated it plainly. Lemma powers differential expression analyses. It's basically the engine running under the hood of countless discoveries. It's that important.
So there you have it. From a sea of 20,000 genes, Lemma helps us find the handful that truly matter. It finds the signal in the noise. And it really makes you wonder, doesn't it? What other secrets are still hiding in our DNA, just waiting for the right detective to come along and uncover them?